In this series of playlists, we have been doing the cell cycle. Firstly, we understood the different phases of the cell cycle. Then we understood what exactly happened in each one of them. We moved on to mitosis, the equational division, and then to meiosis, the reductional division. I'll attach all the links of all these videos in the description and you can refer to them if you like. If you haven't subscribed to my channel yet, let's do it today, and don't forget to press the bell icon so that you don't miss any notifications from Miss Skello. Today we are going to start with the cytokinesis. Cytokinesis is the next step of the division of the cell cycle. So till the telophase, this cell had formed two daughter nuclei that we understood how the division occurs, all the four phases of mitosis and the meiosis 1 and meiosis 2. Then the cell has to divide its cytoplasm, the cell contents and all the cell organelles equally between the two daughter cells, or 4 in case of meiosis. That stage is known as cytokinesis. Now cytokinesis can be equal or unequal. Equal cytokinesis is where the cytoplasm is divided equally between the two cells. You can see this in normal mitotic divisions, and in case of meiosis, it is seen very prominently during spermatogenesis or the formation of sperm cells, where all of the sperms receive equal amount of cytoplasm. However, in unequal cytokinesis, the cell content is divided unequally between the two daughter cells. A very good example of this is formation of the ovum. Now, in a female, when an ovum is formed, as you'll study when we talk about reproduction, the daughter cells do not receive equal amount of cytoplasm. One of them gets about 90% of the cell contents, and the other has about 10. This 90% survives while the 10% degenerates. This 10% exists just to make the chromosome number half. However, how exactly this happens and what are the steps, we are going to discuss when we talk about gametogenesis. These are the two types of cytokinesis. However, cytokinesis also differs between a plant cell and an animal cell. Major reason why this happens is that a plant cell does not have any rigid cell wall holding it together. So a cleavage furrow can easily be formed. The cell can just divide like in case of an amoeba. That's not exactly how mitosis occurs, but we know that during binary fission, the cell just forms a deep furrow and eventually divides. However, in a plant cell, there is a cell wall surrounding the cell. This cell wall does not allow cleavage to occur. Therefore, the plant cell has to develop some new technique, some different mechanism than the animal cell to divide. Cytokinesis starts somewhere between in the late anaphase and ends with the telophase. Now this is variable. This can change from cell to cell, but it's more or less the average duration that cytokinesis takes. Now if you remember, during the anaphase, our cell looks somewhat like this. The chromosomes have aligned in the middle and they are starting to move away from each other. What happens as these chromosomes will now start to move away, the cell has a signal that now the two poles have been decided and it's time to start dividing the cytoplasm so that the two new daughter cells can be formed. As the cell decides to divide, there is a division plane that is set up due to some cell signaling that we don't yet exactly know how it happens, but we do know that there is a division of plane that is set up before anything happens. So as a division plane is formed, a protein known as rho A, which is a GTPase, which means that this protein needs energy to work. It will break down the GTP into GTP, and GTP is like ATP. It functions exactly as ATP does. The bonds this phosphate group store are released, and rho A can then form a contractile ring. Rho A calls out for help. It wants some proteins to come in and form this contractile ring. Two proteins that we know pretty well are contractile are actin and myosin. Here it is myosin 2. And if you know a little bit about muscle contraction, or even if you don't, you'll know that as you use your muscles, your energy is spent, and all of our muscles are capable of contracting and relaxing. Therefore, this contractile ring can contract and it will use energy. The contractile ring will be set up something like this around the cell. Now this is a 2D plane, but you can imagine it in 3D, that it is going from forward to backwards, and in between there is the metaphase plate. As this contractile ring is set, the chromosomes are continuing to move to opposite poles. By the end of the telophase or during the late telophase, we know that the two nuclei have started to form. Now this nuclear envelope is developing, the chromosomes are again condensing back into they are loosening up into chromatins, and our cell has started to divide. The contractile ring now starts to move inwards. It has started to contract due to the actin and myosin. As the contractile ring starts to contract, this entire region that is present here, which is being divided, forms the mid-body structure. Now the organelles are divided equally between the two cells. Here we are assuming that the division is equal. In this mid-body structure are present the cytoplasm, the microtubules that have degenerated here, and all of the structure that will be attached to the middle of the cell. For example, the endoplasmic reticulum that are present in the mid-body structure later form the gap junctions in the animal cells, and these gap junctions are used for transfer of material or communication between the two cells. Now this mid-body structure moves inwards, and as this contraction continues, a final step comes which is known as abscission. This final step is the step where this contractile ring has now completely contracted and the two cells have now pinched off from each other and divided to form two daughter cells, in this case with equal number of cytoplasm. This was the process of cytokinesis in an animal cell that is without cell wall. Now let's look at cytokinesis in a plant cell and how is it different from this cell.